Hello learners, today we are going to learn about the internal structure of a dicot leaf, and we are going to be guided by just one objective that states that by the end of the lesson you should be able to identify parts of the internal structure of the leaf. Alright, as you can see, I've already drawn the internal structure of the leaf and included all the parts that we need to discuss that are related to the internal structure of a dicot leaf. So we're going to start with the cuticle. The cuticle is a wax, and this wax is secreted by the upper epidermis, or the cells of the upper epidermis. Alright, so the upper epidermis is the layer that is found just before the cuticle layer. And the function of the cuticle layer is that it acts as a waterproof. It waterproofs the leaf, meaning that it prevents water being lost through evaporation from the leaf tissues. The thickness of the cuticle depends on the environment in which they are found. These leaves are found. If they are found in the hot environment, you find that the cuticle becomes thicker. If they are found in the environment where there is too much water, you find out that the cuticle becomes thin. Let's move on to the next part. We will talk about the upper epidermis. This is what we call the upper epidermis. And the upper epidermis is made up of tightly pegged cells. As you can see, they are tightly pegged. Alright, the cells that are found on this layer, they are thin and transparent. This is actually an adaptation. There is a reason why they need to be thin and transparent. And also, the cells that are found on this layer, they do not have chloroplast. That means there is no photosynthesis that occurs here. Right. So the function of the upper epidermis is to allow light to pass through. This brings us back to the point in which we stated before that says that the cells of the upper epidermis are thin and also transparent. This is to allow light to pass through. All right, and also another function of the upper epidermis is that it acts as a barrier to pathogens. So no pathogens will enter in the internal structure of the leaf and destroy all the cell tissues that are found there. Alright, let's move on to the next one. We'll look at the palisade mesophyll layer. This is what we call the palisade mesophyll layer, and it is made up of tall, narrow, or you can say long, narrow cells containing what we call many chloroplasts. And we talked about this chloroplast in our last tutorial as the ones that carry chlorophyll, the green pigment that is found in leaves that is there to trap sunlight. Alright, so the function of the palisade mesophyll is that this is the site for photosynthesis. That means that most of the photosynthesis that happens, it happens here. Right, and also it receives carbon dioxide by diffusion. Carbon dioxide enters into the leaf, and then there it reaches the palisade mesophyll so that it combines with water, sunlight, and chlorophyll to produce glucose and oxygen as a byproduct. Alright, we move on to the next part. This is what we call the spongy mesophyll here. The spongy mesophyll is made up of more rounded, as you can see, more rounded cells with fewer chloroplasts than of the palisade cell. Right, so that means here still the photosynthesis also occurs, but not as much as it does here. Right, so being rounded as they are, right, they are loosely pegged. As you can see, there is spaces in between them. They are loosely pegged. So the function of the spongy mesophyll, air spaces between cells allow for gaseous exchange during the process of photosynthesis. carbon dioxide moving to the cells as a reactant, and also oxygen moving from the cells as byproduct. The next part that we can look at is the vascular bundle. The vascular bundle contain what we call the xylem vessel, which is on top, and also the phloem vessel, which is below. Right. The two are separated by what we call the cambium, and this cambium is the one that produces new cells for these vessels. Right. And the xylem transports water and mineral ions, whereas phloem transport food like sugars in through the process that we call translocation. Right, let's move on to the next part. We have the lower epidermis. Here it is. This is what we call the lower epidermis, and it has presence of stomata. Stomata is plural, and in singular we call it stoma. Right, and each stoma is surrounded by a pair of guard cells that are found here. Here are the guard cells, and the guard cells, what they do is that they control the opening and closing of this stoma. All right. Stomata facilitate in transpiration and photosynthesis as carbon dioxide enters the leaf through diffusion, and then also oxygen exits the leaf through diffusion, and this process is what we call gaseous exchange. And remember that oxygen is also needed in plants for the process of respiration. Right? Okay, so this marks the end of our lesson today, learners. Have a good day.